So that it's separate from the lecture recording.
Oh. We don't have to get out and come back in, do we?
No.
Okay.
I mean, you should still be up here. Yeah, you're still in here. All right, here we go. We're gonna get going for the lab. For credit for attending today, you have to participate in the lab Kahoot. Otherwise, you will not be considered having been here today. So be sure that you're signed in. We're gonna get going with this. And again, you can always sign in a little bit of way through. In the Zoom, I have put in the chat. It's at the top of the chat, but it is, retype it in. Kahoot login, Kahoot pin 8098042. In case people join. Okay, here we go. So again, if you were playing through this Kahoot on your own, then good for you, but we're gonna go over way more detail than what the questions have in them. Okay, so for the lab, one of the things, this is part of why everybody needs to, be joining on this, is just because I'm gonna talk about some of the rules for the lab midterm. One of the rules is that somebody just messaged me on the thing and I got distracted. One of the rules is that you have to have your lab coat and closed-toed shoes. If you do not have a lab coat, it will cost you 2.5 points off of your final score to borrow a lab coat. So you must have your own lab coat, not even borrowing one. I'm not allowing that for the test. I'm allowing it, but it costs you points. If you do not have closed-toed shoes, then it will cost you 2.5 points to take the lab exam. I'll let you take it with open-toed shoes, but it's going to cost you another 2.5 points. So be sure you have closed-toed shoes and you have a lab coat. Also, I might test you on some of the lab safety rules. I'm not going to pick weird, obscure rules, okay? I'm going to pick rules like the following. Where should broken glass be disposed? This is a different song for the Kahoot.
It sounds like the original Legend song.
All right, so it goes in, right? It goes in your sharps container, right? So why doesn't it go in the biotrash? Even if it's been contaminated with bacteria, live bacteria, it goes in the sharps. Sharps is a biotrash. It is a biocontainer. And so it counts as a biotrash, but we only put sharp things in the biotrash because we get docked more to get rid of that type of biotrash than with a regular biotrash. So anything that's glass will go in the sharps container because it could break and then cut through the biotrash itself and cause a spill or injury or anything like that. Even intact slides will go in the sharps container. Pretty straightforward starting off. All right, so next we're going to get into the stains. Know the orders of your steps for the stains. Most of the stains are basically the same kind of idea of what the steps are, and it is the following. You typically have stain steps. You typically have a primary stain. The primary stain is the one that really determines the positive or negative. It's the steps after that that are going to help basically like clarify that for us. So it's usually the primary stain, it's the first stain always in the step. First stain is always the primary stain. And this primary stain is usually what's going to determine what the positive or negative looks like. Next step is, if you have it, a mordant. A mordant fixes the primary in place. The next step might be the decolorizer, if you have one. It's going to remove any unfixed primary. Then the last step is our counter stain. And it's just going to stain any decolorized cells. Makes sense? Especially if we look in the context of a gram stain or something like that, this should all make sense of what's going on, all right? So I'm gonna give you a little help with the stains. I said I'm gonna give you, you've already had it. You've had it the whole time. Okay. If you go to modules, this is week 8. If you're ever not sure which week number it is, no worries. Your modules tell you what the dates are for it. So today is October 7th. It says it on my computer. Again, if you don't know it on your watch or something like that, it should be somewhere on your phone, your computer, somewhere. If you don't know what day it is outside of that, I don't know how to help you. All right, so October 6th through 10th, week 8. Right here at the top in the overview, lab midterm study sheets. They've been here the whole time.
There's two of them, A and a B.
That's what I saw too.
Those are not study sheets, those are study guides.
What's the difference?
You tell me, does this not look different than the study guides?
No, it does.
Yeah,
That's why I've been-
yeah. Study guides are questions for you to work through. They're study guide.
Okay.
So I don't know if you guys have been in the past, like people give you a study guide that has just all the information you're supposed to know. That's not a study guide. It's like a knowledge sheet. A study guide is a guide to help you study. So it's like questions about the stuff so you can like test your knowledge and stuff like that. So if you have, so we'll go to that one really quick. I've gone over it before, but I don't mind doing it again because yeah, it's a different thing. So also at the bottom of this week, you'll have lab midterm study guide 1, which will open a new tab, and lab midterm study guide 2, open a new tab. Study guide 1. How can the production of products from plasmids be controlled or turned on or off? How might you tell if your bacteria actually took up the plasmid without DNA testing? What the heck is a Kirby-Bauer assay? What does a clear area around the disk really tell us exactly? Explain the difference between bactericidal and bacteriostatic. If you haven't been working through one of these, then I recommend it. So there's a whole ton of questions like that on lab midterm study guide 1. On study guide 2, read through the lab manual details and follow along with this. Make a chart of the steps for aseptic techniques. Draw depictions of the cell shapes and stuff like that. So this is more like pictures, drawings, visual type stuff, whereas the other one is just questions. They should cover the same ideas, but are you more of an answer questions thinking type of a person, or are you more of a draw a chart type of a person? And you can use either one, and you can use both too, if you're so inclined. Don't let me stop you. But they are written differently, but they assess the same questions. Does that help with that?
Oh, so the lab 1 and 2 are basically the same? They're just.
They should cover the same content, they're just written differently.
written.
Yes,
Okay.
like one
Okay,
asks
that's what I was trying to-
questions and one is like draw a chart.
Okay.
It's based on something. This is 22.
I mean, I wrote them back in 23 or whatever, but like it's the same content that I'm testing you guys on.
Okay.
And it's on there because it's useful, not because it's not useful. I'm not one of those professors that doesn't update my course. This is not that. Yeah. So, no, I get what you're saying. So yes, so this still applies. This is still the same content that you guys get tested over and all of this. Now, what am I going to test you over? You guys already know the answer to this. Lab learning objectives. If you haven't seen this, you're behind, period. Because I've been done talking about this for a long time. So lab learning objectives. We have learning objectives for the lab, just like we have learning objectives for our chapters. So those are there too. So these are the things that I pull my questions from. But notice that we've got a bigger area of a top part. Identify and define key and relevant terms, list the major steps performed, explain the purpose and significance of those steps, and all that. So there's more like, this applies to all of the labs at the top. And then each lab has like more specific information that you'll have to basically know. So these are what I will test you over. Yes. So we still have learning objectives there. So this is new to you. Again, you're late to the party, man. So just saying. So there's that. You can use the lab midterm study guides as a way to test your knowledge for what you've been studying on your lab learning objectives. Okay, so there's the lab midterm study guides. They're a study tool. None of this is required, except, you need to know your learning objectives, just like in the lecture. How do you know what information I might expect you to know for your learning objectives? Your lab manual, quite literally the whole manual, should have all of the information for that learning objective stuff. There's only one deviation from that, and that can be found in your eukaryote worksheet. So the eukaryote stuff that I asked you, we had a student ask a question about it. What do I need to know from that eukaryote worksheet? How was I supposed to answer that? And I said for that eukaryote worksheet, for your transmission methods, as well as your definitive hosts, that you were supposed to look that stuff up for reputable sources, either in your textbook or on reputable sources like the CDC, or if it's a Wikipedia page, it's allowed as long as they have reputable sources that they used. Make sense? And we'll go over that today too. But I have summarized the major learning ideas in our lab midterm study sheets. also all here. It's all here. Your lab manual, your learning objectives, and your study sheets. This is the content part of it. If you want study information for your lab stuff, it's down here. And there's a lab midterm Kahoot. I keep getting emails from people. Where do I find the link for the lab, this Kahoot that we're doing today, right now? Y'all, it's not only in week 8, it's in announcements and everything. Okay, not you guys, obviously. I'm just saying other people from other classes, from other sections. Okay, so it's been there. It's been there the whole time that y'all been enrolled in the class. I didn't add this stuff new. This has been there the whole time. Something else that has been there the whole time, the study sheet plus thing. I'm going to download it because it's a little easier for me to zoom in and stop on this if I have it as a separate file. Okay, lab midterm study sheets. I have each of these study sheets is found in your lab manual at the end of each exercise. So we already had these in our lab manual too, but this is separate from that, just that file with just those things. These don't have everything that you need to know. They have the major concepts that you need to know. I still reserve the right to ask little details about things that might have been related to the lab learning objectives. If it's the learning objectives, you need to know it, okay? But these were my study sheets. I made a study sheet for exercise 2, for example. This one had 2 pages because I needed extra images for exercise 2. But most of the other ones, it's just 1 page. Exercise 3 on 1 page. Exercise 4, acid fast stain, 1 page. Exercise 5, capsule stain, 1 page. These are my 1-page summaries. So that if I was studying for the test, number 1, I made these in my own time. And number 2, if I was studying for the test, I could go back and review this. And it's gonna be more than enough for me to brush up on. But if you're only studying these, you are not getting enough information from them, okay? What I recommend, what I'll say to you and what I've said to my other classes is that I recommend you make your own versions of these. If you're making your lab concept maps for extra credit, if you're making your lab concept maps from ChatGPT and having it write the outline for you, you'll get the points for doing the assignment. But if you're not making it on your own, I don't want to hear it when you get a bad grade on the test. Yeah? I mean, fair enough, yes? So we know the difference between having ChatGPT make it for us and then us doing it by hand. If you're doing it by hand, I want to give all the kudos to you in the world that I can, because good job on that and good job on keeping up with it and whatever. Even if you're typing it by hand, it's fine. But if you're just copying and pasting what ChatGPT made for you, you're going to get your extra credit points, but you're going to need all of those extra credit points to salvage what's going to be left of your grade. You know what I mean? So it's not the same thing to just have ChatGPT do it for you. Same thing with the lecture concept maps, of course, but we're focusing on lab right now, if you're even doing the lab concept maps. But yes, so anyways, this is the same idea as the concept maps. It is a consolidation of the information. The concept maps are prettier in that they have things organized a little bit better. So if you're doing the concept maps, this is the same idea as that. It's summarizing that information in like kind of one place. If you're making 2 sheets for your concept maps, that's fine. But I still recommend it all be as consolidated and easy to see in one place as possible. So this is all here. So these were my sheets. These were all in your lab manual, except for 2 pages. The first of the 2 pages that's not in your lab manual is this one. Exercise 9. It's not in your lab manual because I made you guys do this. You guys remember that? I made you take these pictures. So my pictures, I pulled them from the internet. This, again, I made this like 2 years ago and I haven't updated the pictures in it. So your pictures are more accurate. They're the correct color and everything. But there's only 2 differences between my pictures and your pictures. The first one is aspergillus. Your aspergillus was more of a teal green color, but it otherwise looked the exact same. If we zoom in, it otherwise looked like dandelions versus penicillium, which looked almost exactly like this. And it looked like little paintbrushes, right? These structures are called conidiophores, and I may test you on that. Why? It's been on your sheet that whole time. And I said I could test you on that sheet, right? And it's Unit 1 stuff too. Notice for Giardia and Entamoeba, what do I have written here? Who cares? Not you, because those are not going to be the answer on your test. They might be a choice, but they won't be the answer because they were hard for me to find and harder for you to see them in the microscope. You might have gotten a picture, but because they were hard to see the difference, I'm not going to test you on them. You're welcome. Okay. I'm letting you know that ahead of time. So Giardia and Entamoeba, they might be choices on the test, but they won't be the answer. if we're gonna ask you questions about the protozoa, your protozoas are Trichomonas or Trypanosoma, and please tell me you can see the difference between these two. I feel like it's really obvious that these are different things. Does anybody remember? So here's our Trypanosoma. We see these little snake guys in here. Those are the Trypanosomes. What are these circles? Red blood cells. And we did talk about that in the lab, FYI, but yours should look almost exactly like these. Same colors even, okay? Yours are just a little bit more purple. Your snakes are a little bit more purple. All right. For the transmission route and definitive hosts, I will not ask you about the definitive host on the test. I will ask you about a transmission route. And the question will literally just be, how is Taenia transmitted? Or how is Ascaris transmitted? Or how is Schistosoma transmitted? Okay? And you'll have multiple choice and you'll have to select the right one for whatever one I'm asking about. You will have a question on the test that asks you to identify a fungus. You only have 2 fungi that you had to identify, Aspergillus and Penicillium. You will have 4 choices. I hope that you'll be able to choose between the 2. One of them's green and one of them's pink, for crying out loud. Aspergillus, green, good, gillus. Penicillium, pink. That should give it away already, right? So if it's the pink one, it's Penicillium. If it's the green one, it's Aspergillus. Should be easy questions. Those are the fungi. This section down here, Trichomonas and Trypanosoma, are your protozoa. I might not ask you which protozoa is this. I might ask you what organism is this. And you'll have to know by looking at it that it's Trichomonas or Trypanosoma or Taenia eggs or Ascaris eggs or whatever, okay? You will be asked to identify a fungus. You'll be asked to identify one of the protozoa. You'll be asked to identify one of the eggs by looking at them in the microscope, okay? And you need to know that they are eggs. For example, these were all images of eggs. They were not images of the worms themselves. Clonorchus was one deviant. When on our Clonorchus, it didn't look like this one. Our Clonorchus looked like, do red. Yeah, it was big, right? It was big. It had a shape sort of like this, and it had like this stuff in the middle, dark stuff, right? And that was pretty much Clonorchus. So you know what Clonorchus is going to look like. These are the individual eggs that are inside of this mess. So for what's worth, you'll see this image, that big image. It looks drastically different than everything else, right? I mean, it looked really different. The other deviant was this one. It was bright pink. Pink berries. Yeah, like a raspberry. Right? Bright pink. So that was bright pink. Taenia. Taenia looks like round with round stuff going on inside of it. Ascaris looks like round with like bubbles almost inside of it.
I remembered them by Taenia had the thicker wall and teeth and thicker.
So there's stuff like that. Anything, focus on the differences. Enterobius. These guys were oval and more see-through. Schistosoma, they were oval, but they had spikes on the side. You see these spikes here? All right, so we focused on the differences there. I just wanted to point this out because this has been on our website the entire time that you've been in my class. It's just I didn't tell you about it till now. And maybe some of you noticed that this was here. I doubt it though, but hey, it's been there the whole time. Okay, any questions about that stuff? This is eukaryotes. We're gonna go over the eukaryote stuff in a second, but this is here, this was there. And I feel like it defines a little bit better what my expectations are for the eukaryotes, okay? Yes.
Will these be the only slides that we look at through the microscope Thursday?
Oh, no.
More stuff, more slides?
Like you're gonna have all the stains and stuff like that.
Okay.
Yeah. Yeah, so you'll have the eukaryotes, but you also have all of the stains. I'm gonna ask you questions about how the stains work, what the primary stain is and stuff like that. But I'm gonna put an image, I'm gonna put a slide on a microscope, and the question might literally only be, what is the primary stain used in this procedure? So you need to know, look in that stain, look in that microscope, see it, say, this is purple, therefore it must be a Gram stain. Because none of our other stains had purple cells. Now, one of our other stains had a purple background, but only one of our stains had a background. So if you look into a microscope and it has a background, you've got a reference for it. If you look into it, your stain, if I need to give you more information, like this is a gram stain, what's the primary? I'll give you information as you need it on the test. But some of the questions are literally just gonna be what's the primary stain? Or what's the counter stain? And all you're gonna get is the microscope slide. Okay. There will be other questions on the test, like what is the counter stain for the blah, blah, blah procedure? So you'll get, sometimes it'll be slides, sometimes it'll be just questions about procedures. Okay.
Fill the blank there's-
They're all multiple choice. Everything's multiple choice. And I will say that for the stains too. All of your staining procedures will have very similar choices. So if it's like, what's the primary stain? I'm gonna have listed all of the primary stains. You have to know which primary stain. So I'm not gonna put like bubble de boo stain. I'm not making it that easy. They're all gonna be malachite green, Congo red, crystal violet, harbol fuchsia, etc. And other stains that we might be using as well. Yes. Yes, you do get scratch paper. And you are allowed to brain dump, but again, brain dump, not like from your notes, write down what you need, right? Brain dump. Similar rules to what we have with our unit exam. Remember to bring your laptops. If your laptop doesn't stay charged for an hour, then you need to talk to me about potentially getting a paper test. So an hour should be fine. Okay. Most students will be done within 45 minutes, easy. All right, any other questions about that stuff before we get started with the Kahoot? So this is here. This is your eukaryotes, and there's one other very helpful thing. We've got our DNA stuff, we've got our DNA stuff, we've got our ELISA stuff. We have an appendix page, summary of stains. Simple stain, what's the purpose of it? Do we heat fix? What's the primary stain? Gram stain, what's the purpose of it? Do we heat fix? Primary, mordant, decolorizer, counter stain. What's the positive look like? What's the negative look like? Anything special that we need to know about it? This isn't comprehensive, but it is a good breakdown of the differences between the stains. So as we're going through our Kahoot today, that we're getting ready to start, this is a good reference point if you haven't studied up on this stuff yet, right? If you're doing a brain dump, if you remember the color for a stain, you can write whatever you want on your scratch paper. You're turning it in, right? But you're not reviewing, you're not taking your notes and writing that, gram stain is purple positive, but you're taking it from your brain and writing it on your scratch paper, purple positive for gram stain, whatever, and that's fine. As long as you're dumping from your own brain as your reference, then I don't mind what you write on your scratch paper. Yeah? Okay, so we're gonna get started with the Kahoot. This is here and it's been there, but it is in your modules and it is available for you to refer to for your study purposes. Okay, let's get going. So our stains, again, most of our stains are set up as either primary, then mordant, then decolorizer, then counter stain. If you have, not all of our stains have a mordant, not all of our stains have a decolorizer. So then it's a matter of, does that stain have that? And what is it for that stain? You need to identify what the purpose is of each step in that procedure. So if, for example, your gram stain is crystal violet, then iodine, then ethanol, then safranin. What is the purpose of ethanol? Decolorizer, for example, right? And so questions like that will be asked on the test as well. What's the purpose of each of those steps? And I might ask them out of order, or I might ask them, what is the order and stuff like that. What does a positive look like? And what does a negative look like? Most of our stains, we have 4 differential stains. We have the gram stain, and I know it feels like there should be a lot more, but that's it. We only have 4 differential stains: gram stain, acid-fast stain, capsule stain, and endospore stain. Those were differential stains. We did the motility wet mount. That wasn't really a stain, but was it differential? Yeah. Was it modal or not modal, right? And then we also did the simple stain, which is not differential. It just allows you to have contrast so we could see our cells. We looked at our cell shapes and arrangements that way, right? So know those. And what happens if you skip a step? Some of these steps are pretty important in process. If you skip ethanol in the gram stain, what's going to happen? What would a positive look like if you skip ethanol? What would a negative look like if you skipped ethanol? So understanding those steps and then being able to walk through what would happen if I skipped any of those steps. All right, so we're gonna get into our questions. What is the purpose of heat fixing? I don't know where this music came from. I'm not complaining. Like we're playing Tetris or something, or.
Funny, I was doing this in the dark in my room and I was like, oh Lord, why is this happening so weird? Sometimes they'll change the music for holidays.
Yeah. Maybe it is spooky season music. Alrighty, so purpose of heat fixing is to kill the organism and adhere it to the slide. We did great on this one. So remember, the purpose of heat fixing might be to prepare it for staining, but not all of our stain procedures need heat fixing, and heat fixing itself doesn't stain it. All right, so that's just the difference there in a little bit of semantics. So we're gonna get into this. What is the counter stain used in the gram stain?
Final boss. in the second-
Yeah, this is wild. All right, so in the gram stain, our primary stain is crystal violet. Our mordant is iodine. Our decolorizer is ethyl alcohol or ethanol, same thing. And then our counter stain is safranin. It's the whole reason why our gram negatives are red, because safranin is a red stain, and why our gram positives are purple. Gram positives are purple because the thick cell wall scaffolding traps in that crystal violet. And so it doesn't rinse out, it gets fixed in place. And it doesn't rinse out with the ethanol. But our gram negatives, it doesn't get fixed in place and it gets rinsed out with that ethanol. So we have to counter stain, basically just so we can see the cells for contrast. Typically, counter stains are just simple stains, essentially, just so that we can see the decolorized cells. So they're typically just going to be for that purpose. Another stain type that I want to talk about really quickly. No. We have stain that stains the background and stains that stain cells. Stains that stain the background, these are called negative stains. Stains that stain the cells are called positive stains. They literally have the charge that we're talking about. So imagine this, if you have H pluses attached ionically to a negatively charged molecule, right? The molecule is what is doing the staining. That has a negative charge to it. So this, like we know, if it donates H pluses, it's an acid. So what the staining molecule is negatively charged, these are the acids. These are bases or alkaline. Cells, here's a cell, have a membrane. And that membrane is made up of a lipid bilayer. But what specifically makes up the lipid bilayer of a membrane? That I'm drawing on here. So these are phospholipids, right? You guys know this stuff. So what's a phospholipid? It's a phosphate head attached to a hydrocarbon tail. The phosphate head has what kind of charge? Negative. Has a negative charge. So positively charged things, opposites attract, will be attracted to those phosphate heads. So that's why positive stains stain cells. Negative stains stain backgrounds because negative repels negative. We tracking that? Because chemistry explains everything that you would need to know about this. Just memorizing that positive stains the cells, you're going to get lost in how many cells we're going to talk about in a little bit. So having an understanding of why that's happening will give a better understanding during the test. I'm not going to get that complicated on the test, but I feel like if you understand it at this level, you'll be well prepared for what I'm going to put on the test. Okay, we understanding how positive stains and negative stains work. I'm going to leave that stuff at the side. I'm going to erase this stuff out here. All that beautiful artwork. That's okay. Okay. Move on. Next question. What color will gram-negative bacteria be after the gram stain? All right, they'll be red because of the safranin, right? And safranin stains the cells. So what type of stain is safranin? A positive stain. Beautiful. Excellent. What is the first stain used in a capsule stain? The primary stain for the capsule stain. The first stain and primary stain. Sorry, did you go upstairs? Sorry. All right, what is the first stain used in the capsule stain? Congo red. Congo red. All right, so that is our background stain. So what color is the background in the actual capsule stain? Do you guys remember? It's purple. It's pretty purple, like a bluish purple color, like a big purplish color background. On the picture that we have for the capsule stain, it's more of a gray color because we can get a lot of variation in that color there. But if it has a background, period, it is the capsule stain. No other staining procedure should have a background color, okay? So if it has a background, it's the capsule stain. The background is created by our negative stain, which happens to be Congo red. So Congo red created that background. So why isn't the background red? Because of the secondary stain that we add to it. Secondary stain, that is Manivell's stain. Manivell's stain changes the color of Congo red as well as stains the cells and gives the cells a reddish pink color to them. Okay, so the next question is going to be about Congo red, I think. Yes, what type of stain is Congo red? All right, so it is a background stain, also known as a negative stain, right? So it's Congo red is staining the background, and that makes it a negative stain. Now remember, it's a negative stain, it's repelled by the negative surface of the cells. That's why it stains the background. All right, next question. The image that it's about to show is a pure culture. What stain is pictured? All right, that is the endospore stain. How do we know that that's the endospore stain? Because it's green and red. No other staining procedure is green and red. So when we're looking at the endospore stain here, this is a pure culture, which means that it is one species of bacteria, just one species of bacteria. And these are not pleomorphic bacteria. They don't have many shapes. So the cells are basically one shape, and then the endospores are the other shape. So in this case, our cells actually are vegetative. The active multiplying form of the cells is the vegetative. And the dormant or inert form, dormant, that's an O, apologize. Is the endospore for bacteria, right? So in this image, the rod-shaped guys are the vegetative cells, and the round guys are the endospores. But you see how some of these cells have like literally like a little bit of red, and there's a little bit of green attached to the end of them. These are cells that are making endospores. And those are called sporangiums. And yes, that is in the lab. We did talk about it. Okay. So this is a mixture of all three of those things: vegetative cells, endospores, and sporangiums. The endospores themselves stain green with malachite green. What would a negative sample look like? All red. Positive has bits of green in it. Okay. Our endospore stain, when we looked at ours, so this is pretty greenish. Ours might have a bluer tint to it, but again, red and even a bluish green, bluer green, again, should say endospore to you, right? Especially if we have those different shape structures going on. And if I tell you it's a pure culture and you know that is one, these are not bacilli and cocci. This is bacilli and endospores, not cocci. No cocci in this image. Okay. No cocci here. Bacilli. All right. Any questions about that? This is the #1 most missed question on the test. So that's why I'm trying to kill it with as many stones as I can. All right. You can recognize the endospore stain just by seeing it. It is multiple choice. Yes. And you'll have up to 5 choices. I don't think you'll have more than 5 on any of the questions. Not on the midterm. The final will have more than 5 on some of the questions. Okay. All right. Any questions about that endospore stain image anyways? Yes. Okay. Cool, cool. What is the term for those red structures in that image? The red structures. All right, so those are our vegetative cells. So we noticed we had a couple of other choices over here, the trophozoites and the cysts. Does that seem familiar? Because that should also feel a little bit familiar, because that will also be on the test, is those ideas with the protozoa. So if these are our terms for the bacteria, in pink here, then protozoa equivalent. Vegetative is equivalent in protozoa. Beautiful, I heard you say it. Trophozoite. Do you guys remember how in Unit 2 in Chapter 9, we were talking about all the trophs? And like we were saying about where your energy comes from and where your carbon comes from for autotrophs or heterotrophs or chemotrophs. You ain't going to be trophing if you're dormant. So you're only needing energy or carbon if you're active. So that's how I remember it. It's silly. A troph, those are basically food. And I think of trough and troph food. You only need food and energy if you are active. So our dormant guy for our protozoa is equivalent to the cyst. That'll come back up in a little bit. Okay? So that we know what those other terms are, because you do need to know them as well for the lab midterm. All right. Which of the following bacteria would be acid-fast positive? You can select as many as you want, I think, or is it just one? Well, there's only one right answer. I will say that. So multiple of genuses is genera. And so whenever we have a name of an organism, usually it's the 2 names, the 2-part name, like Escherichia coli or Staphylococcus aureus or Homo sapiens or whatever. That's binomial nomenclature, right? The first part of that name is called the genus. And then the second part is the specific epithet. And the whole thing together is the species name. So the genera that are important for endospore forming guys, these are Bacillus or Clostridium. All right, and then for our acid fast. See ya. We have Mycobacterium. And that's it. So there's only 3 genera that we really need to know for the lab stuff. We need to know it for unit 1 too. Also, you know. So anyways, those are coming back in the lab. Again, so there's those. Mycobacterium, why are they acid fast? Acid fast. Acid fast means that you have mycolic acid wax. Wax is a hydrophobic molecule, right? It's a lipid. So in order to stain these cells effectively, we're going to use a lipid soluble primary stain, which happens to be carbofuscin. Yeah, it could be. So Grace is asking if being acid fast positive could be considered a virulence factor. Could it help with survival? And yes, it does actually, because that wax makes them more resistant to drying out in the environment. So it can actually help them survive better in the environment. So it is considered a virulence factor. So carbofuscin, F. All right, acid fast, F. So that's another way you can remember how that primary stain is for that staining procedure, okay? Mycolic acid, mycobacterium. Just don't fall for the mycoplasma, because that's a whole other different thing. Mycoplasmas are the ones that don't have any cell wall, let alone a thick one with this special wax on it. Okay? Any other questions? All right, what is the primary stain used in the acid fast stain? I gave it away. That's okay. I like the foxes though. Sometimes I just put random pictures just so there's a picture. Okay, so this is carbofuscin. I'm glad we all got that. I would have been very sad if we didn't get that one. So good, okay. Carbofuscin and fuchsia is the color, right? And so the fuchsia, that bright pink color, if you can remember that too, that'll help. All right, in which staining procedure did we use steam? Alrighty, so a little bit of discrepancy here. So this was the endospore stains. We used steam for 10 minutes and we put those drops of the green on top of it with that paper towel that was sitting on top of it. Paper towel was just there to keep everything moist. It didn't do anything else. But the steam acted as a mordant. So it was a physical mordant for the endospore stain. It helped force our primary stain into our capsules. Does anybody remember what the primary stain was for the endospore stain? Malachite green, excellent. So malachite green for that one. And obviously stain those endospores green because of that steam mordant forcing it in there. Good job. Which of the following structures allows for bacterial motility? Select all that apply for bacterial motility. Did it only let you pick one? This is where I try to be tricky, but then at the same time, there's only one answer to this. For bacteria. Okay, so for bacteria, the only structure they can use for motility is flagella. Flagella. For fimbriae are for attachment. The pili are for creating that channel between the cells for us to shuttle the DNA through. And then the cilia are for motility, but they're for eukaryotes, not for bacteria. Okay? Okay, beautiful. So flagella were our ones there. Most of us know that flagella is for motility, but don't get tripped up in thinking there's anything else. All right, so the staining procedures. Simple stain, we're just staining for contrast. It lets us see that the cells are there, otherwise they're clear. Gram stain, we're looking for the thickness of the walls. Is it thick wall or thin wall? And it's usually where we start. It's also our starting point. If you have bacteria, we want to know first, is it gram positive or gram negative? Gram positives that have wax on their surface are acid fast. Gram positives that can make endospores would be positive in the endospore stain. That's our bacillus and our clostridium. So mycobacterium, bacillus and clostridium, all of them are a type of gram positive. All of them are a type of gram positive. So we can put that. These are all subsets of the gram positives. All right. What else? Capsule. Do they have that sticky outer capsule? Remember the functions of the capsule. The capsule is for adhesion, creating biofilms, as well as evading our white blood cells, that phagocytosis. So that was another one that a lot of people missed on the Unit 1 exam. And those concepts will come back up on the midterm because we do cover it in the lab. Wet mount, looking for motility. There's no stain involved in that technically, but looking at motility. Don't forget about what Brownian motion is. Brownian motion is the liquid molecules are moving the cells around. And then the endospore stain. Any questions about the staining procedures before we move on? The next part's going to be ELISA. So I'm going to erase all this stuff. All right, ELISA, first question for it. What does the E stand for? You need to know for the exam what all of the letters stand for, just so you know. All righty, so E stands for enzyme. This is enzyme-linked immunosorbent assay. So we all did great with that. I'm proud of you. Don't get too tripped up with enzyme though, because there will be a question on the test. What does ELISA stand for? And some of the choices, there might be more than one that has enzyme. So just be sure you know, you can recognize the other letters and what they're supposed to be. The assay, enzyme-linked immunosorbent assay. What is an indirect ELISA looking for? So we talked about direct and indirect. Does the patient have the antibody? So I know we at least are in agreement that it should be, does the patient have the antigen or does the patient have the antibody? So this is kind of one of those things. Antibody is made by your body to defend against the pathogen. So that's where gen comes in. I say antigen and pathogen, they both have gen. So essentially the antigen is testing for the bad guy, the pathogen. Does the patient have the antigen? Does the patient have the bad guy? Does the patient have the antibody? Has the body seen the bad guy before and made antibody against it? All right. You will have to know what antibody and antigen are for the test. You will get asked about that. Again, it's multiple choice, but still. All right. And like I said, I'm trying to give you as much information about what's going to be on the test as possible, but typically people perform about the same on the lab midterm as they do on the unit exams. We don't see a ton of improvement on that lab midterm. So it's just a lot of info. And I think that's what it is. People get confused with the colors. So really focus on what the different colors are for the results and what they mean and all of that. All right, ELISA, the principle of how the assay works. It depends on antibody antigen specificity. That antibody recognizes that antigen, and that's what drives the whole thing. And then we have the antibody linked to the enzyme. The enzyme cleaves a substrate and causes a color change. And that's how we actually visualize the results. It's very similar to rapid testing. Rapid testing is the same exact principle, but instead of being in a well changing a color, it moves across a filter paper and changes a line of color. but it's the same exact principle. Indirect always checks for the antibody, direct checks for the antigen. This applies whether it's an ELISA or any other immune test. They always test for one or the other, antibody or antigen. Yeah, I think that covers everything for it. Any questions about ELISA? Okay. DNA stuff, transformation, PCR, gel electrophoresis, and the DNA sizes. Real quick before we get into it, one little mini review for this stuff, because we know that this stuff is also going to be coming up with our Unit 2 stuff. So again, DNA is basically sugar phosphate, sugar phosphate, sugar phosphate. The sugars are linked to nitrogenous bases that are then hydrogen bonded to other bases on the other side. Again, phosphate, sugar, sugar, sugar with phosphates in between. Okay. Phosphate, the P here represents a phosphate, which is PO4. It's got a negative charge. And each of these represents a pair that we call a base pair. Sizes of DNA is based on that base pair. All right, so in PCR, which is polymerase, and what type of polymerase is it talking about? DNA polymerase. Polymerase chain reaction. We have 3 steps in PCR. The first step is denature. Heat separates the strands. The second step is anneal. It's the priming step. We will cool it down and the primers will bind. The third step is the elongate. If the primer's bound, this is the big trick here. Then we will add on complementary nucleotides and build out. So again, walk through the steps. First step, we take a DNA strand and then separate it. The second step, we'll take those separated strands and we will prime them with a primer. It's usually about 30 nucleotides, 15 to 30 nucleotides, but I'm not going to draw that whole thing. And the third step, we've got our primed strands. Zoom, 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 zoom. If the primer's bound, then we can add on and connect in the next guy and the next guy, A to T and G to C, right? And build them out like that. Same thing on the other side. If that primer bound, then we can build off of it. What do you mean?
Elongate.
Nucleotide,
Okay.
nucleotide. A will bind to T and C will bind to G. So this will be in your lab midterm as well as in your Unit 2 exam. So that's why I wanted to take a little time to go over that. So that is the idea of PCR. And lastly, we have gel electrophoresis. It's purple. Gel electrophoresis. We put DNA into a little well inside of a gel that's made up of agarose. Then we add electricity, and then the DNA will move to the positive end because DNA has a negative charge. The big pieces get stuck and the little pieces can move faster, large and small. We following? Okay, so now we're gonna go through the questions. What does PCR do? Right, so PCR amplifies DNA. It just makes copies of DNA. We have a lot of people choosing that it identifies antibodies against the diseases. I'm not sure what my explanation suggested about how antibodies play into this with PCR. But there seems to be a little bit of confusion there. So you might want to double check your understanding about PCR versus ELISA. ELISA looks for antibodies or antigens. PCR is literally just copying DNA strands. Huh?
I chose transfer DNA. I guess I got confused thinking like the nucleotide was like transferring new DNA to the string.
I mean, it doesn't, okay, so you,
Not questioning why I got it.
right, so read your choice again, the one that you chose. It says transfers DNA from one bacterial cell to another. So are we transferring nucleotides to build the DNA? Yes, but are we transferring, yes,
Bacteria.
Yes. okay,
Thank
yeah, you know it. Alrighty, next question. Which DNA fragment is largest of the choices here? Now, easier to see it on your phone maybe than with all my chicken scratch up here. Be sure we're looking at all of our choices and that we know what they mean. Very common question. Which DNA fragment is largest? All right, so 35 kbp because that is 35,000 base pairs. So most of us are getting that. We're not missing the Ks. Definitely don't miss those Ks. So that's what I'll say there. So with that, we know 100 is the smallest, then 1200, then 4, and then 35. Make sense? So go to the next question because next question is going to be about which of the DNA fragments would travel the farthest on an agarose gel. And that's typically how I'll word it. Which one would travel the farthest on an agarose gel? This lets me know that you know how the agarose gel works, and it lets me know that you can identify which ones are the biggest and the smallest. You killed two birds with one stone here. Common question. All right, so this will be the 100 because it's the smallest. The smallest ones travel the farthest because they have an easier time moving through the gel. They move farthest and fastest. Same there. Okay, beautiful. I think that's it for the DNA. Oh, we got transformation stuff. All right, transformation. What type of cells are capable of taking up exogenous outside DNA? Get rid of this chicken scratch now. It's a little tricky for some people, this one. Yes, so this is a little tricky. The proper term for cells that can take up outside DNA will be competent cells. Can pathogens? Some of them can take up outside DNA, but not all of them. Not all pathogens can. Some of them can. Now, some bacteria that are not pathogens can also take up outside DNA. So being able to take up that outside DNA isn't related to being a pathogen or not. It's just bacteria. Remember, there's a lot of bacteria that don't cause disease. They're not pathogens. The one that we used in our example was E. coli. So my empty Tupperware represented an E. coli cell. So that's one example of a type of competent cell that could do that. And not all cells can do that. Only competent ones can. So yeah, so this is just a matter of figuring which category they fall into. All cells are all cells. Some of those cells are competent. Pathogens might be competent or not. And E. coli happens to be competent, one of the types of competent cells. Any questions about that? All right, DNA fragment sizes, the base pair sizes, be able to recognize them, understand gel electrophoresis and why the DNA moves to the positive end because it's negatively charged. How PCR works in a practical sense. Remember that one of the things that I will ask you guys is about if I start with one strand of DNA, it becomes 2, and that is 1 round. 1 strand became 2. In the next step, that two, each one of them becomes 2. So then 2 in the second round, 2 becomes 4. The third round is where people usually get tripped up. Still each of these, and you can draw them out on your cheat sheets, guys. It is fine. If you want to draw them out or if you want to do the math. But in the third round, 4 becomes 8. So don't think it's the third round, 3 becomes 6. That's not what's happening. Third round, 4 becomes 8. And then 8 becomes 16, then 32, 64, and etc. I will ask you up to 6 rounds, and I'll tell you what you start with every time. It's always going to be 1, by the way. I'm not going to make it that hard. Start with 1. How many will you have after 4 rounds, 5 rounds, or 6 rounds? That's what the question will ask. You can draw it out or you can do the math. I don't care. All right, we'll get into the eukaryotes. We kind of covered this stuff in the beginning, but again, that page there that I showed you guys, I wish this would stop. For eukaryotes, this one. This is all you would need to know about the eukaryotes, plus trophozoite, what is it, versus a cyst. Okay, it's on the worksheet, but it's not on this handout thing. So what is a conidiaphore? Well, I don't need you to define conidiaphore, but identify that these are conidiophores, because the lab test might ask what type of structures are you looking at. They're conidiophores. How do we know they're conidiophores? Because they're not covered. They're the spore producing form of the fungus, and it's not covered. If it had a cover over it, does anybody remember what it would be called? Sporangiospores or pores. Sporangio. Okay, so no cover. All right, and then these are the ones that you need to know. Again, I'm not going to put Giardia or Entamoeba as the answer, but they might be a choice. But your answer should be either Trichomonas or Trypanosoma for the protozoa ones. Then you have your eggs for the helminths, and remember that these are eggs too. Okay, so we'll get going with our Kahoot. What organism is this? Based on the picture alone. Pretty similar to what we saw on ours as well. Additionally, if you guys are taking the test and you're ever not sure about a color of a slide, if you look at a slide and you're like, I'm not sure if this is pink or purple, you can ask about the color, and that's okay. All right. I can't tell you if it's positive or negative or what it means, but I can tell you what the color is. All right, so this was pink. This was our pink paintbrushes. This was penicillium. All right, next question. What is the name of this organism? Again, you'll have slides of these. Will be on slides in a microscope. Also, if you ever get to microscope and the image isn't clear, that's not your fault. You shouldn't suffer for that. You can ask me to focus it for you. You can focus it too, if you feel comfortable doing that, but you're more than welcome to ask me to focus it. You're not graded on that. So that was the Trypanosoma. Trypanosoma, it is a protozoa, and that is the trophozoite form of that. All right, so identify those organisms. I say that you had to draw, you guys got to take pictures of them. Identify important structural or reproductive differences, as well as just identify them based on their major differences. Concept of trophozoite versus cyst, know the difference there. And then how the organism is transmitted. I won't ask you about the hosts, but I will ask you about transmissions. All right, now we're on to our general bacteria stuff, media types, cell shape, cell arrangement, as well as isolation techniques and stuff like that. Which of the following is not a term used to describe colony morphology? Definitely for the test, I'm not going to ask you to define the colony morphology terms. I'm not going to ask you to define them. But I will ask you a question on the test, say which of the following is a colony morphology term? And there'll be a whole bunch of sciencey terms, and you'll need to know which one is a colony morphology term. So colony morphology was identifying our stuff on our plate based on what you saw with your naked eye, the macro stuff. What color were the colonies? What shape were your colonies? Was the surface smooth or rough? Was it mucousy and stuff like that? So that was colony morphology. Hyphae is a microscopic thing associated with our molds. So hyphae was the difference here. But convex, rhizoid, punctiform, plateau, irregular. Here's some other ones that you might want to be familiar with. Umbonate, pulvinate, butyrus. These are all colony morphology terms. Okay, and they're on that sheet for exercise 2. Just real quick. These terms. Also, there'll be these terms. This is probably more important ones because they're the ones that I put in the lab manual myself. So punctiform, filamentous, rhizoid, butyrus, pulvinate, umbonate, crateriform. Again, I won't ask you to define them, but you should recognize them as colony morphology terms. Okay, in a lineup. Go away. Okay. My stomach is really growling. Okay. Next, what is the name of this type of culture? These are just like your own black top tubes that we used in the lab. What will we call that? So this season, if you guys got it, but just to be clear, we have some solid media and we have some liquid media. The solid media, there are 3 types of solid media: the plate, the slant, and the butt. We haven't seen a butt in lab yet, but we will in the second part of the thing. The liquid is the broth. All right. These are all solidified by adding agar. Agar is derived from algae. It's a solidifying agent made from algae. The broths are liquid. I don't know what else to say about it. Any questions about the general types of media there? We'll learn about differential and selective media, which I know we covered in Unit 1, but we're going to learn about the different types of selective and differential media in the second part of the lab. It's like that's all it is pretty much. Yeah, and you can always go back and replay this. I don't know how to see the exact answers that you missed, but you can replay this using the link that I posted in the announcement. So it's in the announcement as well as it is a link in your Canvas module here in week 8. So you can use those links to replay it and use it to practice with. Again, I want to be very clear. You guys do realize that none of these questions are like the ones that are on the test. My question, it's not, okay. I think I made that clear with the other tests, but just to reiterate.
Are you talking about like what we're doing right now?
Yes, yeah.
Similar.
So similar types of questions, same concepts, right? But it's not necessarily going to be, I'm not going to guarantee there aren't some that are the same, but definitely I'm not that person that gives my test questions and these are what you need to know. All right. How was this organism isolated? I mean, if I'm telling you it's not going to be on the test, I do mean that, but. Again, we only did one type of isolation in our class, and it was the quad streak plate. The other things on here are also used to isolate things, but we didn't learn about them, and therefore you're not going to get tested on them. I mean, it's quite that simple. So ours was the streak plate, and ours was the quad streak plate. In which quadrant on our quad streak plate do we have the most single isolated colonies? 4. Yeah, 4. Yeah, we put most of our bacteria down in 1, so there's more organisms there, but when they're isolated is when it's going to be quadrant 4, where we spread them out the most. Yeah, good. All right, which arrangement of bacteria do you observe here? Arrangement. All right, so this is the strepto. So overall, this is strepto, which is the chains. Now, Some people look at this and they're like, well, that's kind of bunched, but is that actual bunching or is that just folding back of the chains? And so that would really be the chains. Now on the test, I'm not going to be this, like, I'm not going to give any vagueness. I'm going to say, what is the overall arrangement of what you're seeing here? So the overall arrangement here, we can agree is strepto, right? Chain. And is there some diplo here? Yeah, but what's the overall arrangement? Strepto, okay. What does staphylo look like? Groupings, bunches, yes. They're like grapes, yes. Irregular clusters. You will not, on my test, I only have samples that are either staphylo, strepto, or no arrangement. You will not see sarcina or palisade or really anything like that, okay? So it's really either going to be staphylo or strepto typically that I'm going to ask about. All right. Next question. What cell shape would these guys be? Again, on the test, I'll try to make it as obvious as possible. Almost done. Righty, so that is the bacilli, the rods. And what shape are cocci? Round. And I literally like go by, the bacilli has the L's in it. L's are rod shaped. Cocci, C's and O's are round. So that's how I remember the difference. What's the singular for bacilli is bacillus. So a bacillus is a rod shape. Cocci, the singular is a coccus. Okay, US at the ends of those. Just FYI, that doesn't mean the same thing though. All righty. So colony morphology, be able to recognize that the terms are colony morphology terms. The media types, the solid versus the liquid, and how we solidify it using agar. Isolation techniques, know your quad streak plate, and which quadrant has the most isolated colonies is 4. Cell shapes, cocci and bacillus are all we're going to have on the test. I don't have any other shapes. So know your cocci versus your bacillus, okay? And then cell arrangements, staphylo, strepto, or no arrangement. The different types of agar, like you mean like McConkey agar or all of that stuff, we're going to learn about that. We haven't even gotten there in the lab yet. That was a unit 1 thing. So in the lab, we're going to do that the second 8 weeks of the course. It's like all that. McConkey agar, mannitol salts agar, EMB agar, blood agar, and we're going to do all of these different types of biochemical tests with different types of media. It's all like differential and biochemical and physiological testing of our bacteria with media the second 8 weeks. We're done with stains after this. We'll do 1 more gram stain, but that's it. Okay. Before this lab midterm, just knowing like liquid and solid medias and what they're called will be enough for now. Okay. Again, you're not going to have, we haven't used the butt yet. Therefore, the butt is probably not going to be the one that you're going to see on the test, right? So FYI. But we will have butts and we're going to stab them with needles, okay? All right. Microscopes and how to use them properly. This is pretty straightforward stuff too, I feel like. If you observe a specimen at 40x and you have an ocular lens that's 10x, what's the total magnification? I feel like we had a question like this on the unit 1 test too. So by now, most of us know how to figure this one out. All right, so we will multiply them, and all of us got this one right. Congratulations, I'm actually pretty proud of you guys for this one. I think you're the first class that I've had that got all of them got it right. So we'll multiply 40 with 10. So anytime you multiply by 10, you just add a 0 on to the other number. For those of you that might not be great with math. But yes, and so I will have a microscope that'll be set up and it'll be on a slide, and it'll be asking questions about the slide too, but somewhere in there it might ask, what is the total magnification, how this microscope is set up and how you're viewing the image through that microscope. And what that means is you'll look at the ocular lens and you'll look at that objective lens and you'll multiply them together. Our ocular lens is always 10x. So that's good, okay? So whatever that objective lens is on, if it's on the 40, it'll be 400. If it's on the 100, it'll be 1,000. Beautiful. Good. Right. Which objective lens do we use for oil immersion? For our microscopes, this will be the 100X. The 100X has the oil immersion with it. And what's the purpose of oil immersion? Yep, reduce the light scatter, also known as refraction. Okay. Be able to calculate magnification, understand how the microscope works. I'm not going to make you guys, like I said, I'm not going to have a microscope and be like, set it up for me and focus on the thing. I don't expect you guys to be experts at that, okay? But what I do expect you to know is what the parts are called and how they work. So one of the things that I want to talk about with this, in this image that we have over here, okay? We have, dang it, this black piece right here, and it's the same as on ours, right? It's the part that it's the rubber piece that you use to rotate between your different lenses, okay? It has a name. Does anybody know what it's called?
Selected.
Nose piece. I heard it from somebody. Who said it? Okay, good. All right. I wish I could give extra credit for that sort of stuff because yes, it's the nose piece. It's the nose piece. These are the objective lenses, right? All right. So if those are the objective lenses, that black piece has a name. It's called the nose piece. The nose piece rotates between the different objective lenses. Now, I'm not going to tell you which test. It's going to be either this midterm or the final where I'll ask about the nose piece. And people get it wrong almost every time. And then they get mad at me because I'll have a piece of tape on that rubber piece. And I'll say, what is this part? And another question is, what does that part do? Well, if you didn't know that that is the nose piece and that it rotates between the objective lenses, and most people, well, they'll put like that's the objective lens. No, if I meant the objective lens, I'll put the piece of tape on an objective lens, not on that black part above it. So the black part has its own name. It is a nose piece, okay? So FYI. Now, when you have a microscope and you're using it, the light travels out of the light source here through the condenser, which is this next part here, condenser. And then we can open and close the condenser using that switch on the front, the diaphragm, right? Then the light comes out of that and it hits the specimen here and it goes up into your objective lens, right? That objective lens, we get our first image made. Which image is that one? The real, excellent, real image, because that's the first image that we're making. Then it goes up through some like mirrors and stuff like that and out through another lens here. This is the ocular lens. And then ocular lens is inside of the eyepiece. Then here at the ocular lens, we will be making the virtual image. We good? Any questions about that? I think that we're pretty solid on microscope stuff. So yes.
I'll ask about like little slider, like that iris.
The iris, so that slider thing, the switch on the front is called the diaphragm. And then that hole that the light comes out of is called the iris. So that's all part of the condenser. Yes. Mm-hmm. And then it's in your little notes thing too. I have a pretty good picture of a microscope in that one. Okay, Kirby-Bauer, this is our last one, all right? Now, just looking at this Kirby-Bauer plate here, I would say that we would be, where am I saying? Just pick a different color, let's put red because I feel like it's pretty easy to see. That this one's susceptible, this one's susceptible, 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 resistant, resistant. This one may be resistant. I can't really tell if that's growth there or not. If it might be susceptible or resistant. This one is definitely resistant. I know it has some of a clearing, but it's still growing inside of that clearing. It's not good enough, right? And then this one is questionable. It's hard to see. I'd probably say susceptible. And then this one, I think it's still growing inside of the clearing, so I'd probably say resistant. Now, this is just base value. On the test, I'll be sure if I ask you about something like this, that is obviously clear or obviously grown over. I'm not going to pick any weird middle of the road gray area, okay? But this is just my face value assessment of our Kirby-Bauer assay. Now, if I take a sample inside of this clear area here. Shoo. And I put it on a new plate. If it grows, or if it doesn't grow. If it grows, then that means that that antibiotic, LZO, whatever it is, antibiotic, is exactly bacteriostatic. Static. If it didn't grow, then LZO is bactericidal. Can you tell that just from looking at this plate here? Nope. We know that it didn't grow, but we don't know if it killed it or if it just didn't grow. Is it static or is it cidal? We can't tell from that first plate. We have to do the second plate to determine that, right? Okay. Another thing about Kirby-Bauer, what's that media called? Do you guys remember the media that we use for the assay? It is called Mueller-Hinton agar. It is not called Kirby-Bauer agar. It is called Mueller-Hinton agar. And it has a very controlled pH and a controlled depth. These controlled pHs and controlled depth help control antibiotic diffusion and stability. Not for the bacteria, for the antibiotic to diffuse and be stable. So also so that we can compare it from plate to plate and stuff like that. The clearing area, whatever this is, I can AMC or something like that. If we measure this, what is this called? The zone of inhibition. Zone of inhibition, the zone where we inhibited the growth, right? And we can actually use this zone of inhibition. We didn't calculate it, but you could use it to calculate the minimum amount of that drug that you needed to inhibit the growth, which has a name. It's called the minimum inhibitory concentration. All right, I think that's everything for Kirby-Bauer. Any questions about Kirby-Bauer? All right, we got one question for it on the Kahoot, and we are done. Which antibiotic is this bacteria least susceptible to? Don't overthink it. Alrighty, so it's gonna be E. And so I see some of you picked, zoomed out for D.
Yeah, this is versus.
Yes, so if you are resistant to something, you're not susceptible at all. So we're least susceptible to the thing that we're resistant to. So don't rule out susceptibility there. So again, what we will say in the Kirby-Bauer, you are either resistant or susceptible. I can't today. Again, susceptible and sensitive. If you are resistant, you are not susceptible, but then therefore you have zero susceptibility. That's why E was the correct answer here. Okay. Next would be D, but don't rule out the E because we're not susceptible to it at all. That's a zero susceptibility. We have some susceptibility in D, but E is a zero susceptibility. If you're susceptible, you are either bacteriostatic or bactericidal. So those are, again, back to our three possibilities. If you're resistant, we didn't do static or cidal with it. Okay. All right. Any questions about Kirby-Bauer still? Now that we've done this. All right. So last thing, delete, clear all drawings. Zone of inhibition, minimum inhibitory concentration, Mueller-Hinton agar, bactericidal, bacteriostatic. I think we've covered it pretty well. All right. So that is it for this Kahoot. Happy studying for it. Really quickly, for the lab midterm review, if we took the test today, Our average would be a 54. To put this into perspective, okay? So y'all sitting there thinking like, this is easy stuff, we're trucking right along. Our average would have been a 54 if that was your exam, and that's with me walking you through everything. You see what I mean? This is what I'm saying. I think it's easy stuff too when I'm just walking through it with my professor. But your exam is in 2 days. Not even 2 days, right? So keep that in mind. It's about 2 days. Keep that in mind. If you got studying to do, you got studying to do. You can do better than this, and you will do better than this. And I know you will. Go through this stuff. And remember, the goal is to get to where you can explain it back to me. If you can explain it back to me, you are more than prepared for this stuff. Okay, focus on those learning objectives, focus on the lab manual content, and then work from there. All right, here's our little podium. Congratulations. You guys are going to do great. You can do this. You've got this. You've got still 2 days to prepare. Get your studying down. Email me with any questions that you might have. Guys can do great. You will do great. I know you will. And I'll see you on Thursday for the lab midterm. Okay? Good job, guys.
I do have a question.
Yeah, what was your question?
Oh, never mind. It's a dumb question because I just answered it myself. Never mind.
You can email me too, so don't worry about it.
No, I got it. I'm in La La Land.
Yeah.
If it's susceptible, that's when you're gonna have. Some side. Bacteria stack. But if it's resistant.
Might have to be an option.
All right, we're doing it. You guys got this. You're gonna be awesome. Yeah. I mean, I hope so. I think it is, but
You gotta put on like live.
I have, yeah.